Hi, everybody. So thank you so much for allowing me to do this as a recorded session. My name is Eman Chaudhri. I'm a fifth-year medical student, recent graduate of Al-Faisal, and for those of you who may not know, but I have taught you guys before, so you guys most likely know. So today I'm going to be teaching you guys hearing physiology specifically. So we're going to be going over the anatomy and the physiology of the ear. But more specifically the inner ear today, because we're going to be talking about how the brain perceives hearing, how hearing actually occurs, and ways, for example, that sound and volume of sound is modulated within the ear. So I'm going to be teaching you guys this with Dr. Yaqeenuddin's lecture itself, because it touches upon everything that you guys need to know. And inshallah, it'll be easy and quick. I hope to finish this within 45 minutes, but if I don't, I'm sorry. And if you guys see me get up in the middle, it's just to turn on the light because the sun is probably going to set within the next 30 to 40 minutes. All right, so let's get started then. Now I'm just worried that if I close this or put it in presenting mode, it's not going to show. So I'm going to keep it like this, and we're going to look at it this way just to ensure, because usually when I put on presenting mode, it doesn't work sometimes when sharing screen. All right, so hearing. There are multiple parts of the ear that are involved in your hearing, from the outside to the inside of your ear. And what's interesting is each part has a very different process or like function, and they're all important in order for sounds to properly reach your cochlea, which we will talk about. The cochlea or vestibulocochlear apparatus meaning like, all those structures in one area, are all there in your inner ear, and they're responsible for the perception of sound, and the vestibule more specifically for balance. However, they do go hand in hand, and I'll show you guys that in a moment on how that happens. But before we get into that, let's just go over a few, different details about hearing, different things about volume and sound, etc., so that we can go into that then. So this is what we're going to need to know for this lecture by the end of it. So we want to know the function of the outer ear, middle ear, and inner ear, and understand the mechanical structures and why they are, that way, what they're contributing basically to sound energy that is transmitted eventually to your cochlea and auditory receptors. And then we want to draw the human audibility curve and explain the changes that occur with aging, explain the frequency analysis performed by the cochlea on the basis of its physical properties, explain how deformations of the basilar membrane are converted into action potentials in the auditory nerve fibers, diagram the auditory pathway with all the central connections, so basically in the brainstem up into the cortex. Describe how pitch, loudness, and localization of sounds in space is coded by central auditory neurons, and distinguish conductive central and sensory neural deafness. So what we're going to be focusing more on in this lecture, for what I want to explain to you guys, for you guys to really understand, is the cochlea itself, the apparatus generally, and the structures within the cochlea, because that is more important for your guys' understanding of hearing, and that relates to the what is it called? The basically the curve that they show you to show basically hearing loss in different people, whether it's sensory neural or conductive. Sorry, I forgot the word for a second. But anyway, continuing on, because all of those things are connected. And if you really understand the physiology of the ear and why, for example, the cochlea and hair cells act in a certain way, then you're going to understand what happens in hearing loss. All right, so hearing. So sound is the oscillation of molecules. So when we speak, or well, let's just say you're listening to music and you're hearing it through a speaker, I do this sometimes. I'll kind of put my hand, like, for example, here's my speaker. So above the speaker, this is where the sound comes out from, especially if it's loud. I don't have to be touching it, but you will feel a little bit of air kind of touching your hand. And that is because it's the oscillation of molecules in the air. Sound travels through the air, and specifically in sinusoidal waves like this, right? This is how it goes. So when you have a low sound, it's going to be like this, smaller waves. If you have a louder sound, it's going to be like this. If you have a higher pitch, it's going to be closer like this, and a lower pitch is going to be like this. So the wavelength is what's different in the pitch, but the amplitude is what's different in the volume. So make that connection. Amplitude, volume, wavelength, pitch. Okay? So hearing is our perception of the energy carried by sound waves over a range from 20 to 20,000 hertz. And we're going to go over this again when we look at the cochlea and the structures within the cochlea, because hearing within this range of hertz is important, because if it's outside, then your hair cells basically, or structures in your cochlea, don't actually get activated, or they don't get acted upon, which you guys will understand in a moment, and therefore you won't be able to hear it. That's why certain animals can hear pitches of sound that we cannot, and we can hear within this range. Now, the amplitude of the sound, as I mentioned, is a total change in pressure, and it will basically be shown and depicted in the loudness, the volume. So higher amplitude, louder; lower, quieter. Now, the wavelength, if it's closer together, it's a higher pitch of sound. If they're farther away from each other, that is a lower pitch of sound. So the change in pressure produced by sound waves is a sound pressure amplitude. And why this is important is because it will activate more or less of your cochlea, whether there's more pressure or lower pressure. And also, it's important to understand this in the context of pressure, because the pressure within air is different from the pressure within liquid, which is in your cochlea. So over here, in our atmosphere, this is all air, but within our inner ear, it's actually liquid or fluid. And so the pressure is different, and pressure also has to be modulated, because too much pressure can cause damage, too little pressure, no sound, right? So the unit for the sound pressure amplitude is done in decibels, and you guys have heard of decibels, and you don't really need to remember this or know this. It's just more for the sake of understanding, but even then, this has never come in a previous exam. You don't really need to understand anything about any calculations or anything like that when it comes to hearing or for balance, okay? So don't even worry about that. So then, let's go to the next slide. So peak sensitivity, or ability to hear quiet sounds, and auditory acuity both lie within 1,000 to 4,000 hertz. So even though you can hear from 20 to, let's see again, 20,000, the more typical and basically, I guess, favorable range is usually between 1,000, or according to other people, 1,500 to 4,000 hertz. So this is the frequency range for human speech. Women's voices are typically a higher pitch, men's are a lower pitch. And sound transduction turns airwaves into mechanical vibrations, and that's what we're going to talk about later. So within the cochlea, we have multiple structures. I don't want to talk about it here in word form. I would rather show it to you in a diagram. Okay, so we'll go into that in a second. So this is an important concept for you guys to know. Because when we talk about hearing, because we're becoming clinicians, the reason that you guys are learning about hearing is not just to understand like why hearing occurs. It's to be able to, for example, for those of you who actually want to go into otorhinolaryngology, it's a beautiful specialty, by the way, aka like ear, nose, throat, ENT, that specialty. You will have to know how to conduct a Rene and Weber test, which we're going to get to at the end of this lecture. But for you guys to understand why we do that, you guys need to understand how waves basically travel through air and what happens when they're not traveling in a certain way, or if they're traveling and the ear apparatus or apparatuses, I don't know the plural, in your inner ear cannot pick up on that. So when you have a tuning fork, so give me 1 second. Okay. If you all right. So I brought my tuning fork so I can show you guys properly. So away from the lecture for a second. To activate a tuning fork, we usually just hit it against like a bony process. And if you guys heard that, so right now it's vibrating. And what we do is we either put it on the bone or we put it next to the ear, and you can hear it. For a tuning fork, they're not going to ask you about this now. They might. You usually see, and you'll see it right over here, 512 or 1024. This one is a 512. Typically, this is what you're going to be seeing in the clinic later when you guys go in 5th year to do ENT during your surgical subspecialty. So when you have a tuning fork and you do this and you hear it, basically you feel the vibration, and that vibration is traveling in what are known as concentric waves. So remember what I said about hearing or sound being in sinusoidal waves like this. Concentric waves are like circles, concentric, like you could think center circle, etc. So they're traveling like this. Imagine them like pushing the air, and imagine sinusoidal waves traveling through the air at a distance. Okay, think of it that way, because vibration you can feel, but literal sound or hearing, you don't necessarily feel that. However, they do come together because when you're speaking, again, sound is the oscillation or movement of molecules through the air. All right. So the frequency ranges for different organisms, there's a few different frequency ranges. And as we spoke about, for humans, it's 20 to 20,000. For cats, it begins slightly higher and goes even higher. For bats, it's the highest, and moths and mice. Elephants, it goes at a slightly different range than humans. It goes even lower. And for dogs, it's more or less the same. We don't really need to know this, but it's just for you guys to understand the fact that frequency differs from organism to organism. And there's a reason that we can hear within a certain frequency, especially in between 1,500 to 4,000, whereas for other organisms, they hear different ones. Now, to the anatomy of the ear. So you have the external ear, your middle ear, and your inner ear. So your external ear, what you can literally touch and feel, and usually where you're cleaning earwax out from, hopefully, that's all your external ear. And you have this area, or let's just point to it over here since my scarf is covering my ear. This is known as the pinna of the ear. And this is basically controlling the sound that comes in. It also helps with localization of sound. And moreover, this entire area of the external ear and the way it is physiologically and anatomically, it is protecting your tympanic membrane. Like, for example, look at the distance between the actual outside and the entire external auditory canal, all the way to your tympanic membrane. It's for the protection of your tympanic membrane and also to maintain a certain temperature and moisture for your tympanic membrane to work properly, because dryness and other environmental factors can possibly damage your tympanic membrane otherwise, and you wouldn't be able to hear as properly, and you can possibly get conductive hearing loss. So over here, your sound will travel through and it will hit your tympanic membrane, and your tympanic membrane will kind of move back and forth when it hears a sound, depending also on the volume of the sound. So the amplitude of the sound waves that hit it, the oscillating sound waves. There's also concentric sound waves, which is more from vibration. And these then, well, you'll have your malleus, incus, and your stapes. These are your middle ear bones, okay? Your ossicles. So the ossicles will move in a certain way because they will detect that the tympanic membrane is moving, and there's tiny little muscles in your middle ear attached to these ossicles or these little bones that will kind of make them turn in a certain way and do so to protect the tympanic membrane and also to transmit the sound to the inner ear or the cochlea. And we're going to talk about that more in a second, specifically how they move, how those muscles move the ossicles and how the sound actually gets to your cochlea and why it gets there in a specific way and it doesn't just travel directly through. So again, the role of the outer ear, it will allow the sound to be localized and then funneled through from the pinna into your external auditory canal, which is here. Pinna, external auditory canal to your tympanic membrane. And also, again, protect your tympanic membrane and the delicate parts of the middle and inner ear from impact. This is all protective mainly, and it heightens your sensitivity to sounds. Now, the middle ear we talked about, there's the bones and little muscles connected to the bones, right? The bones you need to know are the malleus, incus, and the stapes. The malleus is connected directly to the tympanic membrane, as you can see over here. This is the malleus, this is the incus, and this is the stapes. So the malleus is at the tympanic membrane. So you can remember it this way: membrane, malleus, membrane, malleus. Then the stapes is at the window, the oval window of the cochlea or the vestibular apparatus in your inner ear. So remember malleus, membrane, then it's the incus, and then the stapes. You can kind of think of this as the stapes kind of looks like, you know, the end of the saddle. So it all looks like a saddle, and it's like where you put your foot. So you can imagine that you sit here and then this is where your foot goes, if you want to imagine that. I don't know, for some people that was helpful before. So the malleus, incus, and the stapes. These are all your ossicles or your ossicular chain. And what this does basically is they will attenuate sound, and they're involved in your attenuation reflex. That is an important term that you guys need to remember, attenuation reflex. And it transmits the sound from the external ear, obviously, to the internal ear. It's right in the middle. So what else is it going to do? But specifically, what we need to understand about the ossicles and about the attenuation reflex is how they actually are working in context of when sound is traveling through. So let's just say a sound travels through and it hits your tympanic membrane, and your tympanic membrane starts moving. Your malleus is attached to the tympanic membrane, right? Now, you have a muscle called the tensor tympani that is attached to the malleus. So this is the malleus, and this is the tensor tympani. It's going to pull on the tensor tympani is going to pull on the malleus in a way where instead of the tympanic membrane being able to move like this, it will stay put like this. It becomes taut. Taut is a way of basically saying kind of tight. And the reason that happens is because it wants to reduce the amplitude of the eardrum so it can protect it, so that your eardrum doesn't burst or it doesn't get damaged or it doesn't transmit too much sound to the inner ear, because that is also detrimental to the structures in your inner ear in the cochlea. And we will get to that in a minute. I know I keep saying that, but this all kind of builds upon each other because it goes through. Sound travels from out to in. So we have to talk from out to in. So think of the tensor tympani acting on the tympanic membrane. So remember, membrane, malleus, tensor tympani, tympanic. So all those three structures, the tensor tympani, malleus, and the tympanic membrane act together, okay? Then remember that you have the stapedius. And remember that you have the malleus, incus, and then the stapes, and then over here is your vestibular apparatus and cochlea, basically your inner ear. So at the stapedius, so at the inner ear, you have this oval window over here where your stapes is. And your stapes, which is the little bone that is attached to the oval window, is attached to a muscle called the stapedius. So stapedius, stapes. You can see how these kind of go together and it's easier to remember. So the stapedius will basically contract and it draws the stapes more outward, so it reduces the impact on your inner ear. The organ of Corti is a structure within your cochlea that we will talk about. And why this is important is because basically when you hear a very, very loud sound, let's just say you're at a concert or you're at a wedding or a gathering and people are playing very, very loud music, what happens is that your stapedius is kind of contracting so that your stapes is also outwardly contracting a little bit or moving outward, so that your organ of Corti or your inner ear is not receiving all of the sound that is coming from your external auditory canal. And why that happens is because it is protecting the organ of Corti, which has your hair cells, which is in the cochlea, and the hair cells are responsible for transmitting sound, or basically sound as electrical impulses to your brain, right? And if your hair cells are damaged, then you can't hear. So when a very loud sound is transmitted, you have this attenuation reflex, which is where your tensor tympani and your stapedius muscles are contracting in order to move the ossicles a certain way to make the tympanic membrane taut and to move the stapes outwards so that the sound is kind of, stopping. The amplitude is not as much of the tympanic membrane, and also so the stapes can move this out and the organ of Corti here stays safe, okay? This is all happening within 40 to 80 milliseconds. So it's very, very fast. SubhanAllah, it's really, really cool. And it's very important for the protection of your middle ear. Now, impedance matching is another concept that's pretty important. So impedance is any obstacle in the pathway of sound and expresses the sound energy lost as sound passes through any medium. So when we look at sound, again, as sinusoidal waves, and it's traveling through air, sound when it's traveling through air is traveling at a certain speed or at a certain, you can say, in a certain way. But when it changes a medium, so for example, when it goes from air to liquid or fluid, which is in the inner ear, it's not going to travel as fast. So what happens about that? That's not good, right? Because you want to be able to hear. If you're hearing a certain volume, you want your inner ear to be able to get, the sound at that volume. But that's not going to happen if, none of these structures were there and the sound is trying to travel from air to fluid. It would just stop. And your hair cells wouldn't even have enough frequency to pick up on in order to actually be able to do anything. Your hair cells within the cochlea. So sound has to be conducted from an air-filled middle ear to a fluid-filled inner ear. So let's go back to this. This is air-filled and this is all fluid-filled. So from air to fluid, obviously the pressure is changing, right? And the frequency is going to be changing as well. It's not going to be the same, and you have to take care of that because you need your hair cells to be activated so that you can send electrical impulses to your brain to perceive the sound that you're actually hearing. So there's acoustic resistance And the pressure is increased at the junction between the middle and the inner ear at the oval window. So what happens basically to take care of this problem? This like natural problem that is always there for every single human being. Well, the ossicles in chain, so the malleus, incus, and the stapes, they act as a lever system and they increase the force of movement about 1.3 times. So they're acting in a way to transmit the sound. Remember, they're doing their own thing. They're moving kind of like clockwork in order to change the way the sound is being perceived from air to fluid. And more than that, the surface area of the tympanic membrane is greater than the stapes in the oval window. Although the same force is applied, but the pressure is increased by 17 times at the face plate of the stapes. So the pressure is increasing as you go in. And again, like I said to you guys, you don't really need to remember anything about numbers, but this is just for you guys to understand that there are pressure changes from air to fluid, and the ossicles are what are important and what are responsible for impedance matching. So instead of there being impedance, it's basically matching what is coming from the air to the fluid, okay? They're moving the not the stapes. They're moving the tympanic membrane and acting on the oval window a certain way in order for the sound to be able to go from air to fluid in a natural way where the sound waves don't necessarily decrease in frequency or anything, and they stay the same so that your inner ear can perceive it correctly. Now, if anything happens to any of these structures, from the tympanic membrane, any of your ossicles, basically in your middle ear or external ear, this is known as conductive. This is known as a conductive problem. Remember, this is conduction. It's going through. It's trying to conduct the sound to your inner ear. So that's what we call a conductive problem or conductive hearing loss, which we will get to in the end, and we'll tie everything together. Now we get to the beautiful cochlea. It is such a tiny and delicate structure in your inner ear, and the way it acts is so cool. Not to sound like a nerd, but we're going to talk about it right now. So let's zoom in a little bit and let's move the slide if I can peacefully. Nice. Okay. So you have your cochlea. I'm going to show you this photo. Wait, first. Actually, let me show you guys a better photo. Just for better photo, I mean, for the sake of introducing this concept to you. You see? All right. So over here, if you guys can see this image, it says Ken Hub over here. I hope you guys can see it. I hope it's sharing correctly. That's the only downside of recording. I can't have you guys to give me feedback in case you guys can't see anything. But over here, you'll have your vestibular apparatus, you have your oval window, and you have your cochlea over here. Now you'll see this thing in between, kind of this like membrane or line, this basically barrier, right? This is separating two portions of your cochlea, and see how it travels here from the base, spirals down to the apex, okay? And you see how it doesn't end over here. There's a space over here that allows for the movement of the fluid. That's very, very important because this, I will explain in a second, but this allows for the movement of the fluid, and that will allow for certain, for example, pressures to be maintained in the cochlea, and I'll show you how in a second. But just remember that it's a spiral, okay? But for the sake of understanding the physiology and, you know, in relation to the structure, but more for the sake of understanding the physiology, imagine that you're kind of, you know, imagine this is like a fruit roll-up and you're rolling out the fruit roll-up and you're seeing it as one kind of like strip now, okay? Rather than like a spiraled, rolled up thing. So I'm going to reshare this. So this is the cochlea. It's cut in half. Your fruit roll-up, remember, your spiral, it's cut in half. And this is what you're looking at. So we were looking at it cut differently. You saw that barrier, that line. If you go over here, oops, this is that same barrier or line. And this is actually the basilar membrane. Now, when you have sound that travels from the external ear to the middle ear, and now, you know, all that attenuation reflex, impedance matching is done. We've talked about that. And it gets to the inner ear. Your oval window, obviously, sound waves are traveling through. And what happens is that will cause movement of the fluid in your inner ear. What is that fluid in your inner ear? Now look over here. You have your oval window that we talked about. This is your vestibular apparatus, etc. This is your cochlea, and this is your vestibule. Now, your vestibule has these semicircular, semilunar canals, right? And this is your cochlea as a spiral. Your cochlear, the scala vestibuli, basically the scala vestibuli, scala media, and the scala tympani, the portions of your cochlea are filled with perilymph, okay? Perilymph. Today, we're going to be talking about perilymph. However, the vestibules or the semicircular canals are filled with endolymph. So just make that, just distinguish between those two because they're different. So it's perilymph for your cochlea and endolymph for your semicircular, semilunar canals. So let's talk about the perilymph. So your oval window, stapes, etc., the sound travels through. Your perilymph then is going to start moving in a certain way. And remember that the perilymph is very, very rich in potassium. And that's important because you have certain cells, and remember that cells depolarize, repolarize, etc. And what happens in depolarization, repolarization? What is it in relation to? Potassium and sodium, right? So that's why that's important. You're going to have potassium in your perilymph, and that's very rich in your perilymph. Now, let's look at it at this angle because this is important for how the sound will travel. So over here, this is your scala vestibula. Scala basically just means duct, right? Scala media, so media you can think middle, and then scala tympani. Now, the perilymph moves in your scala vestibula, or the vestibular duct of your cochlea, okay? And when this happens, this vestibular membrane, also known as Reissner's membrane, basically is activated, and it will send or move the perilymph basically in your scala media. Remember, it's all filled with fluid. And now let's see what happens. So let's look at the structure over here. You have your hair cells. This is all the organ of Corti, and this is the tictorial membrane. So these contain your hair cells, okay? And what do your hair cells do? Why are they important? Well, your hair cells, basically, they're called hair cells because they have these little projections that are like hair-like projections, okay? Your stereocilia, they're called stereocilia. So stereo, you can think sound. Cilia, you know what cilia are. They're on cells and they're projections. So when you have the movement of your perilymph, obviously, let's just say you have, your cilia and you have fluid, they're going to move back and forth, right? You can think of it kind of as like a sea anemone, like in Finding Nemo. They move back and forth basically in water, so in a fluid, right? So when they're moving back and forth, what happens is they're opening certain channels at the tips of the hair stereocilia and the kinocilium, which is the biggest stereocilia, okay? So I'd like to find you guys a bigger diagram of that. So remember what I just showed you guys about this. This is your organ of Corti, and if we zoom in, take this basically and magnify it, you have your hair cells, your afferent fibers of what? The vestibulocochlear nerve. And these specifically are contributing to which portion of the vestibulocochlear nerve or cranial nerve 8? The cochlear portion, okay? Because it's for hearing, not for balance. So you have your hair cells, your afferent fibers, then you have your tictorial membrane. And the tictorial membrane is basically kind of rubbing across, pretend my fingers are the stereocilia, across the stereocilia because they want them to move in a certain way. And why? Well, let's look at this diagram right here on Google that I just found. Right here. I hope you guys can see this. But basically, I just showed you the tictorial membrane and these are your stereocilia. This is a much, I guess, like simpler made diagram. But they're going to move back and forth because of the movement of the perilymph, right? And the perilymph is going to move them in a certain way because at the edges, they actually have these channels. And what happens with these channels? Firstly, they're connected by cadherin proteins, okay? Second, so that's why when one stereocilia moves, the others move. And why? Because when they all move, all those channels at the tips are opening. And what happens when those channels open? Let's look at this. You see these channels? So this is your stereocilia. This is your kinocilium, the biggest stereocilia. Basically, what happens is potassium, remember, which is rich in the perilymph, is moving into the cell along with something else, calcium. And what happens when we move calcium into the cell? Remember, if you guys remember from neuro, and I'm sorry, from POD, I'm sorry to bring it up, your calcium will basically go to where your vesicles are stored near the end of a cell, because sometimes those vesicles are storing neurotransmitters, because what's happening over here? Sorry, guys, the Zoom account logged me out for a second. But basically, what's happening over there? So over here, you have your afferent fibers, right? That are going to the cochlear portion of your vestibulocochlear nerve. So when the potassium and the calcium is like rushing in, and that calcium is there, remember what happens with the vesicles? They're able to go and synapse to the synapse and release all the neurotransmitters. And in this case, the neurotransmitter is glutamate. So the glutamate will go and it will bind to the to the ends of your neurons, and it will send an electrical impulse through your afferent nerve fibers into the cochlear portion of your vestibulocochlear nerve, which then travels through the olivary nucleus up to your brain and everything, and that's localization of sound and other, higher functions that are being done by your brain, and that's a perception of sound, ultimately. Isn't that so cool? So it's basically just from sound coming here, moving the fluid in your inner ear, your hair cells picking up on that, moving in a certain way for them to open the channels to allow calcium and potassium into the cell to allow for depolarization and for the calcium to go to the vesicles and allow those vesicles to go to the end and release the neurotransmitters into the synapse, and for those neurotransmitters, aka in this case glutamate, to go to the ends of your afferent nerve fibers of your vestibulocochlear nerve, specifically the cochlear portion, and allow for an electrical impulse to be sent through. And that is what will cause the electrical impulse then to be sent to your brain. So this is explained here in a little bit more detail. But let's first now review all of that from the beginning to the end, specifically for the cochlea, right? So we have our organ of Corti, like we talked about. We have our outer hair cells and our inner hair cells. So one thing that I actually forgot to mention for over here is our inner hair cells are responsible for the sound, but our outer hair cells are responsible for mediating how much of the volume we're actually listening to. And this is a protective mechanism. All right. So you have your hair cells that are basically going all the way, remember, let's look at the cochlea again. All the way from here, from the base to the apex. Now, what's important about this is at the base, that's where you hear the highest frequencies or the higher frequencies. And the reason for this is it needs the highest frequencies or the higher frequencies that you're listening to be able to move, because it's a stiffer part of your cochlear membrane or of your cochlea. The apex is a little bit more floppy and membranous, and so that is where the lower frequencies are heard. So we're going to be talking about something called presbycusis later, which is like the loss of hearing, especially related to old age. It's age-related hearing loss. But the first sounds that they stop hearing are the higher frequency sounds or noises. And that is because those are the first hair cells that are exposed to sound. And therefore they get damaged first. So they stop hearing higher frequency sounds first. So you can always make that connection. Higher frequencies at the base, it's closest to the outside, it's the first to be lost. And the apex is more floppy. It's for hearing lower frequency sounds, and it's the last to be lost. We'll talk about that again so that it really, really sticks in. All right. So you have receptor cells in the organ of Corti that are specialized, that are two specialized nerve cells called hair cells. So hair cells are basically a specialized type of cell, therefore a specialized function, specifically a special sense, basically a special sense, hearing, such as like taste, smell. Hearing is also a special sense. All right. So you have your outer ones, like we said, they're responsible for mediating sound, and the inner hair cells are responsible for the actual hearing, for the noise, and they start moving as soon as sound waves are perceived. Okay. So the bases and the sides of the hair cell synapse with the spiral ganglion of Corti, which sends axons into the cochlear nerve. So what this means is these afferent nerve fibers that I was talking about over here, you don't see it over here, but there's a spiral ganglion, okay? And that spiral ganglion is just something that has afferent and efferent nerve fibers. That's why it's a ganglion, right? So the afferent nerves, obviously, afferent is going to go, because it's sensory, to your brain. So then where is the efferent? Like, what is it sending back? Remember, I just mentioned these outer hair cells. So these inner hair cells, sure, they're going to be sending things to your cochlear nerve. Now, these outer hair cells are responsible for mediating the sound for the sake of protecting the structures in your inner ear. But how is the brain supposed to know that you're hearing too much or the frequency is too high? It's more than like necessary, and therefore your ear needs protection. The brain needs to send back signals in order for your hair cells, your outer hair cells specifically, to move in a certain way. So the outer hair cells control the sensitivity of the inner hair cells at different sound pitches, okay? So they will send back, basically, their efferent nerve fibers, which will release acetylcholine, and that will cause certain proteins in the basilar membrane to cause the hair cells to move down away from the tictorial membrane, away from all of this, so that it's not perceiving as much sound, and therefore it is protective to your inner ear, to your hair cells. It's going to move this entire apparatus down, okay? Now, I watched a Ninja Nerd video earlier, basically to review hearing physiology and anatomy, and I highly recommend that you guys watch it. I already put the link at the first title slide, and he goes into more detail about this if you guys want further understanding, even though it's not necessarily going to be tested, but this is just for your understanding. It's better for you to know so you understand why it's happening. So sound transmission, let's do this again really, really quickly. Remember, you have your sounds, oscillation of molecules, etc., through the air, sinusoidal waves, concentric waves, etc., blah, blah, blah. Okay, cool. It's going to your tympanic membrane. Your tympanic membrane is vibrating. Your malleus, tensor tympani muscle, pulls on the malleus to make your tympanic membrane taut so that the amplitude does not stay too high, and therefore it controls the amplitude of your tympanic membrane and it doesn't damage it. Furthermore, it starts basically rotating with the incus and the stapes. And the stapes, with the stapedius muscle moving the stapes, it will move it outwardly so that it's protecting your organ of Corti, or your inner ear, basically your cochlea. And your organ of Corti, again, is running through your cochlea. And the sound waves, again, remember there's impedance matching and the attenuation reflex in your middle ear. So the ossicles are moving in order for the attenuation reflex. So what we just talked about, the protection of your inner ear from loud sounds, and impedance matching, making sure that the waves are moving smoothly from air to fluid, because remember that the pressure changes. So your sound waves are going to go through, and it's going to move your perilymph in your scala vestibula. Vestibular duct, scala means duct, so vestibular duct of your cochlea. Now remember what I said over here about that barrier and how it doesn't go all the way to the end. There's like a little space over here where, it doesn't connect. That's because your perilymph is moving from here to here into your scala tympani, so the tympanic duct. And why does that happen? Remember, sound enters from your oval window, travels, and goes to your round window. Now, why does that happen? You need to control the pressure of your inner ear. Simple as that. Remember, when the sound waves are passing through, they can't just stay there, because if they stay there, then the pressure is going to increase and increase and increase, and then destroy your cochlea and destroy your hair cells once and for all, and you're never going to be able to hear again unless somebody knows how to transplant new hair cells, right? So what happens is the sound waves are going to go through, move the perilymph, and then exit through the round window back into the air. And this allows for the pressure to stay basically equal and normal at all times. And that's basically what you need to understand about that, okay? So the sound waves in the air strike the tympanic membrane. Sound wave energy is transferred to bone of the middle ear, which then vibrate. And the vibrations are transmitted via the oval window to the fluid within the vestibular duct and create a fluid wave within the cochlea that we talked about. And that fluid wave starts in the vestibular duct, or the scala vestibula, then goes to your scala tympani, or tympanic duct, out through your round window in order to maintain the pressure. Okay. So now when you have this difference in perilymph movement, you're going to have deformation of the cochlear duct, and that causes the tectorial membrane, which is this part right above the hair cells, to move and activate the stereocilia. So what does that mean? Again, like we said, if these are my stereocilia, like I mentioned before, this is the tectorial membrane, it's moving them back and forth. Specifically, it's going to move them in a direction. And if it moves, let's just say they're normally like this, and if it moves them like this, now the channels at the end are open, and what happens? The calcium and the potassium that are rich in the perilymph rush into the cell, okay? And the potassium works for depolarization, and the calcium works to get those neurotransmitters, specifically glutamate, out of their little vesicles into the synapse to go and get the afferent nerves from these inner hair cells, afferent nerves, to the cochlear portion of the vestibulocochlear nerve and travel up to the brain. I hope that made sense. And that is the entire idea of how the cochlea works, okay? So you have your scala vestibula, scala media, which is, in relation to the vestibular membrane, aka Reissner's membrane, and your organ of Corti, and you have your tympanic membrane. So remember, the perilymph is moving from your vestibular duct to your tympanic duct, out, then to the, sorry, not the perilymph. The sound waves are moving through your perilymph from the vestibular duct to the tympanic duct and then out through the round window in order to maintain the pressure. Now, this is another diagram that I found also helpful. If you guys might find it helpful too in a drawing form from the Ninja Nerd video. So it's the same idea. This is your stapes, looks like a little, part of the saddle, your oval window. The vibrations from these from your little, bony ossicles in your middle ear are traveling to transmit the sound waves through your perilymph in your scala vestibula, SV. Scala vestibula. Your vestibular membrane separates your scala vestibula from the scala media, where your tectorial membrane is. And that perilymph movement will cause the tectorial membrane to move across and kind of like brush and push the stereocilia on your inner hair cells. And your inner hair cells, like we said, potassium, calcium, all of that, potassium will cause depolarization, and the calcium will cause the vesicles to release their glutamate and go to these afferent neurons as neurotransmitters and cause an electrical impulse to be sent in the afferent fibers of the vestibulocochlear nerve. And the spiral ganglion, which we cannot see here, well, it's over here, covered by his hand, has afferent and efferent nerve fibers, right? The afferent, like we said, they're going to the brain, and the efferent are going to go back to these outer hair cells, because in case it's too loud or in case the frequency is too high, it's going to say, hey, listen, relax. We need to protect the ear. Move away from the tectorial membrane and move away from this membrane, basically. All right. And that happens by the release of acetylcholine from these efferent nerve fibers. So afferent glutamate, efferent acetylcholine. So it'll move away from the reticular lamina, as you can see over here. Now, sensory coding. Remember what we said about the base and the apex, and how the base perceives higher frequency and the apex is floppy, it perceives low frequency. This is basically just saying that in more words, okay? So think of our fruit roll-up rolled out now. I'm sorry for making that relation. I know people don't like to relate food to human body structures, but it's going to happen a lot. Your fruit roll-up is rolled out. So here, this is your external ear, your middle ear, you see your ossicles over here, your malleus, incus, and your stapes. And you see this is where your oval window would be. This is your vestibular duct. This is your tympanic duct. Now, this is your entire cochlear. Imagine that it's like rolled up, right? This is where the higher frequencies are heard, and this is where the lower frequencies are heard. Now, this end or the apex of your cochlea is known as the helicotrema. That's just the name of the structure. Okay? And now this happens again, like I mentioned, the basilar membrane of the cochlear duct is stiff and narrow next to the oval window, and it becomes wider and more floppy or flexible near the distal end, further away near the helicotrema. So the location of the hair cells in the membrane creates a code that the brain translates about the pitch of the sound. So your brain knows when the hair cells are activated here, that's a lower frequency sound and your brain can pick up on that. When it's over here, it's a higher pitch sound. So for example, children's voices, women's voices are typically more here. All human voices are generally considered higher frequency or higher pitch sounds, right? Over here, but maybe a man's voice is over here, slightly lower pitch. So that is typically why, again, in age-related hearing loss, you're going to have loss of higher pitch noises such as children and female voices first, as compared to male voices and then the other sounds. So it's the same idea over here. Near the oval window, you hear higher pitch. Near the helicotrema or the cochlear apex, you hear lower pitch. That's what they're tuned for. So it's tuned for higher near the oval window, basically at the base, and the apex is tuned for lower pitch. And this is the same thing again. And this is the transduction of hair cells that we were talking about. Remember that there's a membrane, all cells have a potential. And what we were talking about was the movement. These cadherin proteins connect your channels together that are on your stereocilia. And then excitation is when they move this way, and then inhibition is when they move back. All right. So this is what we were talking about. And maybe it would have been more helpful if I had put this slide above for you guys to see it while I was explaining. But movement of your stereocilia. So you have depolarization, which is when the tinctorial membrane moves your stereocilia and then the potassium enters in, and so does calcium. And these will go to your vesicles and release neurotransmitters, which in this case is glutamate. Okay, so what happens is the potassium will enter through here. Specifically now, I'm going into a little bit more detail here now. Potassium will enter through these when the stereocilia moves. So the channels on your stereocilia are for potassium, okay? They will move in, and this will cause depolarization. And depolarization opens voltage-gated channels. Remember, it's an electrical impulse. So these voltage-gated channels are responsible for calcium. So the calcium then will enter into the cell, attach to these vesicles, and allow them to open at the basal side of the cell and release their neurotransmitter to go to the afferent nerve. All right? And then again, that's the afferent neuron of your vestibulocochlear nerve, cranial nerve number 8, up to your brain, and the spiral ganglion, which is right there under. And the spiral ganglion will also send back efferent nerve fibers to your outer hair cells to control the pitch and how much is actually being perceived by the hair cells in order to protect them in case it's a very high frequency or a very high pitch. Pitch and frequency are interchangeable terms in this case, okay? So this is another way to look at it. Your vibration in the basilar membrane, displacement of the hair cells, the stereocilia, opening of the transduction channels, influx of potassium that causes depolarization of your hair cells. You have opening of your voltage-gated calcium channels and calcium influx that causes synaptic release of glutamate, response in the cochlear nerve, and therefore then it goes to your brain. So now we've said it about 4 or 5 times. So hopefully, inshallah, it should be clear. And I see that Dr. Yaqinuddin also used Ninja Nerd. And now, since you guys understand everything that happens up until it reaches the nerve fibers, now let's actually talk about where the nerve fibers go, right? Because it doesn't just end there. It's going up to your brain and it's being perceived. So you have your auditory pathways and they travel through your olivary nucleus, okay? Your olivary nucleus is here in the medulla. Then it goes up, goes to the nucleus of the lateral lemniscus, and then up here into the inferior colliculus, and then into the medial geniculate body, and then up to your cortex. And this is what allows for you to organize, basically spatially, where you're hearing the sound from, to organize frequencies and pitches, to understand them, and to also localize. And localization doesn't only happen as a higher function, but it also happens with the use of both ears. You can localize, is the sound coming from behind me? Is it coming from in front of me, above me, below me, etc.? And remember one thing, because you guys did just finish neuro. The left cortical area is larger than the right and is closely associated with Wernicke's area. Remember, Wernicke's area, you have Broca's and Wernicke's, which is here. So Broca's is frontal, Wernicke's is like temporal parietal, right? Temporoparietal. So the Wernicke area is responsible. Remember, when we have Wernicke's aphasia or Broca's aphasia, let's talk about Broca's first. It's just a speech problem, right? Forget about that. But now this is related to hearing, Wernicke's area. Remember what happens when we have Wernicke's aphasia. They have fluency, but we don't know what they're saying. They're just going to be like saying a bunch of random words, which they think makes sense to them, but it doesn't make sense to the other person. So that also has to do with hearing. So this is another way to look at it. So the cochlea is, again, inner ear. You have your auditory nerve fibers. They're coming from your inner hair cells, going through your medulla, specifically the olivary nucleus, traveling through your brainstem, going to the inferior colliculus, medial geniculate body of the thalamus, because remember the thalamus is responsible for all of your sensory input, right? It organizes all your sensory input. And then it goes to your cortex, specifically to the primary auditory cortex, where Wernicke's area lies. This is another drawing of Ninja Nerd as well. So we all know, so olivary nucleus, lateral lemniscus, inferior colliculus, medial geniculate body or nucleus, and then cortex. So for you guys to remember that in case you guys do get tested on it. Inner hair cell, spiral ganglion, olivary nucleus in the medulla, travels up lateral lemniscus or nucleus of the lateral lemniscus, inferior colliculus, medial geniculate body, and then up to your cortex. Okay, so write that out basically as like arrows. And this is another way of looking at it. I guess it's like a more of a drawing, but we're not going to go into this. So many sound features are encoded before the signal reaches the cortex, right? So remember, sound localization, understanding pitches, categorizing them, pattern recognition in sound, in localization, etc. These are all higher functions and they're all done in the cortex, okay? So you have descending cortical influences and they modify the input from the medial geniculate nucleus. This is important as an adaptive filter for the noise and for the sound that you're hearing. So remember when we talked about the outer cells, that's basically where the function happens through. So your brain is basically sending that to your inner ear and telling your outer cells, okay, move away from that membrane. It's too much pitch or it's too loud, or we need it for the sake of protection of the hair cells. So that's another way of describing it. All right. So this is further telling you more about this. So there's like some longer neuronal paths, some shorter. And there's the action potential will travel from your inner ear all the way up to your brain. And then also, again, there's afferent and efferent neuron pathways, and it's important to understand between the two. So this is explaining more about what happens inside your brainstem. Now, everything that you guys need to know is already written on the slide, and I don't think that there's anything more that I really need to build upon. So you guys can go over this yourself. If you guys have any questions, you can let me know. This is the last slide, and now we will quickly, within the next 10 minutes maximum, hopefully, talk about tests and deafness. Let me just quickly turn on my light because it's getting a little bit dark. All right. That's much better. Okay. Because I want you guys to be able to see this. So deafness obviously is an issue, and you need to be able to measure deafness because deafness can occur due to different things, or loss of hearing. Loss of hearing, I think, is better to say. So you can have conductive hearing loss or sensory neural. So think about those words yourself and you can know what they mean. Conductive in the conductive path or in conduction, in the middle ear or external. Sensory neural, sensory neuronal. So it's in your inner ear or in the nerve fiber itself, okay? Now, conductive hearing loss can be any problem that is, causing any issue in your external or your middle ear. And there's different causes. So number one is the most obvious and most common one. Earwax impaction, cerumen impaction. And a lot of the time, when you remove the cerumen or the earwax, when it's been impacted, and why does it get impacted? Usually because of those Q-tips, right? People use Q-tips and without realizing they're not cleaning their ear, they're actually pushing the earwax further in. That impaction can prevent sound from actually hitting your tympanic membrane. So people might mistake that as like, oh my goodness, I can't hear in one ear, what's going on? So that is usually the problem, primarily. Otitis media, or inflammation or infection of your middle ear, usually it damages the tympanic membrane in the ossicle. Sometimes your tympanic membrane can rupture, and it usually will heal on its own within 1 to 2 days. But it can also damage your ossicles, or it can cause a lot of, fluid buildup, and therefore sound cannot travel properly because the vibration cannot be conducted through your bones properly if there's so much fluid there and if there's inflammation, right? And the other thing is pathological fixation of the stapes to the oval windows, osteosclerosis. So obviously, if you can't move the stapes freely, and if you can't move it with the oval window, then how are you going to transmit sound properly as sound waves into the perilymph? You're not going to be able to. So these are all conductive problems. Now let's look at nerve deafness. There's multiple issues, or there's multiple causes of nerve deafness. And this is either due to cochlear problems or auditory nerve problems. Now there can be degeneration, which is age-related, like we said, presbycusis, which we'll talk about at the end. It can be hereditary. It can be toxic due to certain medications, such as, for example, if you're taking, you guys are going to learn this in CVP right after summer, inshallah. There's certain diuretics that you take in heart failure that you have to warn the patient that if they start having hearing problems, it could be due to these diuretics and they should go to the clinic then. And those are known as loop diuretics because they act on potassium. And if you're reducing the potassium, then what happens? You're reducing the potassium in your perilymph. And if you don't have potassium in your perilymph, then what happens? You can't depolarize your hair cells, right? It all connects, and it's really cool when you get how it connects. So it can be due to toxicity from certain diseases or from certain medications. It can be trauma, acoustic trauma. So you hear a very loud sound, such as people, for example, who are in combat. If there's an explosion or a bomb, acoustic trauma can occur. It can be due to meningitis, because remember, meninges, cranial nerves, it's all covering and it all travels back. So meningitis can cause problems in your auditory nerve. And acoustic neuroma. So you guys will remember from your neuroscience block, when you have a vestibular schwannoma, or if you have an acoustic neuroma, for example, I think the same, if I'm not wrong, they're the same thing. Maybe they're interchangeable. Correct me if I'm wrong, please. It's been a while since I've done neuro. That can actually impede how the sound or how the electrical impulses travel through your nerve. Okay, so that's also a sensory neural problem. Now, we talk about Rene and Weber's test, and you guys have probably heard about this, and you guys will definitely be practicing it in pro. But what happens is in Rene's test, you're comparing bone conduction to ear conduction. So you take your tuning fork, you make sure it's vibrating, you hold it from here, okay? Do not hold it from here because you're going to be stopping the vibration. You hold it from here, put it on a bony prominence and get it to vibrate. And you're going to put it near your ear, and typically they can hear. This is air conduction, This is air conduction. You can put it on the bone here, and you can usually feel a vibration going through. And if you can't really do it that well with a scarf. Typically you have to take the scarf off. But basically, when you can hear it going through the bone, that is bone conduction. If you can hear it in the air over here, that is air conduction. So now you can differentiate between conductive and sensory neural hearing loss with the simple test, right? So normally your air conduction is always better than your bone conduction. And think about it, naturally, when you're in your environment, air conduction is always better. You're hearing sounds coming from everywhere, they're coming through the ear. It's not like people have to touch the bones behind your ear for you to be able to hear them, and then somehow their sound is being transmitted that way. It's being transmitted in the air, right? So your air conduction normally is always better than your bone conduction. But let's just say a person is not able to hear. Let's just say they're like, doctor, I can't hear in my right ear. I don't know what the problem is. And you do Rene's test and you do this. And you put it over here. They can barely hear it. Sorry, 1 second. Alhamdulillah. Let's just say they can't hear. They can maybe hear it or very little. But let's just say when you put it at their bone, they're like, yes, I can really hear that properly. Well, what happened right now? Instead of this sound, the sound waves being transmitted from the outside in the air to the middle ear, being conducted through your ossicles and then going to your inner ear, all naturally, if they can't hear this, but their bone conduction is better, that means they probably have a conductive problem because you had to put it in the bone, which, by the way, because it can then travel through your skull bone to your inner ear and not through the air, because there's a conductive problem. Now, if you have a sensory neural problem, then both your ear and your bone, it doesn't matter because your nerve is a problem. Everything can be taking the sound there, but if there's a problem in the apparatus and how to perceive that sound, then that's a sensory neural issue. But when the bone conduction is better, if they say, doctor, I can't hear this, but I can hear when you do this, then that means that there's a conductive problem because it couldn't conduct itself from the outside to the inside. You had to conduct it here from the skull bone to the inner ear. So the sensory neural portion was fine because it was able to perceive the sound, right? Or the sound waves. It was able to go about all this process that we've been talking about for the last hour. But it wasn't able to get the sound there on its own. You had to conduct it on your own. So this is conductive. If the bone conduction is greater than the air conduction, it is a conductive hearing loss or conductive hearing problem. If both are reduced, it's sensory neural or mixed. Some people can have mixed conductive and sensory neural. Now, let's talk about Weber's test. This is really interesting. So we do the same thing. But now instead of putting it here, we're putting it in the middle. Now, normally, you don't hear it, or you can hear it in both. You're like, yeah, okay, I can kind of hear it in both. It's going towards both. Normally, majority of people will say, because we just took this in ENT, that you don't really hear it. You're like, I don't really notice a difference. But sometimes people can hear it better in one ear. What does that mean? Now it's different in between conductive and sensory neural hearing loss. Remember what we said, conductive, the bones, the external ear, everything middle ear could not take the sound to the inner ear. In sensory neural, the sound can get to the inner ear, but the inner ear doesn't know what to do with it because there's a part of the inner ear or the nerve that is not functioning, right? So let's just say we do this. Now let's just say this person says, oh, doctor, I'm not really able to hear that well in my right ear. We don't know the cause, but this will now tell us, along with Renee's test, right? So we do this. Let's just say we put it in the middle, and they said that they couldn't hear that well over here in their right ear. But they say, oh, doctor, when you do this, actually, I can hear it better in my right ear. What does that mean? I thought you just said you couldn't hear in your right ear. Well, let's just say you did Renee's test as well. And their bone conduction was better than their air conduction. And when you did Weber's test, the sound was localizing to their right side. What does that mean? Sound will localize to your working inner ear. Remember, your inner ear wants to pick up sound. It's literally there. That is its purpose in life. It wants to get sound and it wants to perceive sound. So your inner ear is fine, but for some reason, when you did the air conduction, it wasn't really getting but when you did the bone conduction, it was better, right? So again, when you do this, it's conducting through bone. And because your cochlea is all intact and the nerve is fine, then that means that, okay, the nerve is fine, the cochlea is fine, but the reason that you can't hear is maybe because of a conductive issue. So in conductive hearing loss, when you do Weber's test, the sound will localize to the affected ear. But now let's just say, doctor, I can't hear in my right ear. And when you did Renee's test, their air conduction and bone conduction were both decreased. And when you did Weber's test, the sound is localizing away from the affected ear. What does that mean? Remember what I said about sound. It wants to go to the healthy cochlea. It wants to go to somebody who will listen to it. Remember, we want people who will listen to us. The cochlea want to listen to something, okay? We want the cochlea that will listen to us, the cochlea that is healthy. So it will lateralize away from the affected ear. So if you see that in a vignette, patient comes in complaining that they cannot hear in their right ear. And when Weber's test was done, because sometimes they won't tell you that both Renee and Weber's was done. Sometimes that's going to make a question way too easy. But Weber's test was done, and the noise or the sound lateralized away from the affected ear. That means this cochlea is healthy, and this cochlea was affected. They couldn't hear it over here. That's sensory neural hearing loss of the right ear. However, doctor, I can't hear in my right ear, and you did this test, and the sound is lateralizing towards the affected ear. It's affected in conduction, but not in the cochlea, because remember, the cochlea wants to hear it. Now you're asking, okay, if it is conductive hearing loss in the right ear, why is it that when I do Weber's test, it's not lateralizing to both sides if they're both healthy cochlea? Well, This cochlea is all alone. It's not perceiving sound, and it's been in a quiet environment for so long. This one, remember, even though you can hear me talking, for example, you're still having your environmental sounds around you, right? And your environmental sounds are also being perceived by your cochlea. But this cochlea is not perceiving any of those other environmental sounds, so it has no one else to disturb it. So when you do this, your cochlea is like, oh my God, yes, finally, some noise. It's going to lateralize to this cochlea because there's no other environmental disturbance to affect the perception of sound in this cochlea. As simple as that. So that is why unilateral conduction deafness, it will be louder in that ear. That's the third point on the slide. Whereas sensory neural, it will lateralize to the other side because this cochlea is healthy. Remember, cochlea want to hear your noise. The cochlea are literally waiting for sound waves. So if this has been waiting for sound waves for longer because there's a conductive hearing loss, it'll lateralize to that side. As simple as that. I hope that makes sense. So over here, this is basically talking about that. This is from first aid, this table. Swiber's test, which is where you put it in the middle. Normally, there's no localization. Sometimes people might say like, oh yeah, I hear it equally in both, but not really. Typically, you don't hear it in either. Conductive, it's going to localize to the affected ear because there's less transmission of the background noise. There's no one else to disturb it. Sensory neural, it's going to localize to the unaffected ear because that cochlea is working. It wants to hear your noise. Renee's test, as I said, normally for all of us, because normally when we're talking and perceiving sound is coming from the air. Air conduction is always better than bone conduction. But when bone conduction is better than air, that's a conductive hearing loss problem. And if both air and bone conduction suck, then that is a sensory neural problem or a mixed conductive and sensory neural hearing problem. Presbycusis, what I've been alluding to this entire time. This is the gradual deterioration of the spiral organ of Corti. So you have your organ of Corti and you have your hair cells, as we talked about. Presbycusis is a loss of hair cells over time. It is the gradual loss of hair cells over time because of degeneration, okay? And it can be due to trauma over time. Remember, when you're hearing more things over time, it's more likely to have damage over time, okay? So typically, this is in patients who are 65 or older in age. And typically they will complain first again of hearing loss of higher frequency. So most likely they will say, you know, doctor, I kind of avoid going out with my friends, especially when they want to go out to like a restaurant or somewhere where there's a lot of people because I can't hear them and it's embarrassing. And I keep saying, huh, like I can't hear you. And they feel embarrassed, like they don't want to go out with these people then. Sorry, my clip is falling out. And other times they might say that, you know, my wife will be talking to me, but I can't really hear her, or my children will be talking to me, but I can't really hear them that well anymore. That's because those are higher frequency or higher pitch sounds. You lose your higher pitch sounds because, again, they are at the base and not the apex. The apex is a lower frequency sound. So those hair cells are typically the last to get destroyed in presbycusis, in gradual hearing loss. Hearing loss, again, is progressive and cumulative, so you can remember it that way. And with that, we're done. So thank you guys so much. I hope it was all clear. And if you guys have any questions at all, please feel free to text me. Please feel free to DM me on anything, email me. I'm happy to answer your questions. I'm actually studying ENT also because my final exam for surgical subspecialty involves these concepts. So if you guys have anything you want to clarify, let me know, or tell people in PAL to that you have a question if you don't know how to get to me directly, and they will let me know. So thank you guys so much, and I hope you guys have a good time in the rest of HNS and studying for your exam. Bye.